from Manipal University, India. Let me be very honest, Dr. Arun T.S., the first author, has actually carried out the work. I'm one of the co-investigators. Professor Gopalan Kutty and Professor Mallikarjun Rao are the guides and co-guides respectively for this work. So the title of the work is this: Metformin interaction with popular Indian herbs, Boswellia, serrata and Withania somnifera, PKPD approach with normal and insulin resistant model. Now, in India, different systems of medicine coexist with modern medicine like Ayurveda, Siddha, Unani, herbal medicines. So it may not be an exaggeration to mention that every Indian household uses herbal medicines, be it in the form of a food constituent or a herbal preparation, because the general notion is that herbal medicines are safer alternatives. And often they are taken concomitantly with allopathic medicines. So there is a general perception that they are safe, and that leads to, because they are safe, that leads to concomitant usage of alternate remedies. Now, what was the need of this study? If you take product monographs, there are drug-drug interactions reported in a product monograph, but herb-drug interactions go underreported. So that was the need of the study, because we have seen from one of the papers, Adrian Berman's paper, where he has reported that many people who suffer from chronic illnesses, along with their allopathic medicines, they mix a concoction of herbal medicines because they have comorbid disorders, and most of those drug interactions have deleterious effects because they either increase or decrease the effect of the drug. So that results in underreporting. So that was the need of the study. Therapeutic benefits or toxicity from the alternative medicines can not be underestimated. There are many drug interactions. They can be antagonistic interactions that render the drugs ineffective, synergistic interactions that can enhance the effect, or it can be toxic, drug-drug interactions, herb-drug interactions. So one of these is possible. Then there is another case where deliberately drug interactions are intended. Now, why is this? That is to modulate the PK or the pharmacodynamic profile of the drug, to provide an improved therapeutic response, or to minimize the ADR profile. So the need of the study, it was very relevant. As I've already said, in India, there are vast population which suffers from comorbid conditions where, along with allopathic medicines, they take other medicines too. That goes underreported. And most of them ail from chronic conditions. And the effect results in polypharmacy, scarcity of scientific drug, especially for herbs. There is no particular monograph which details herb-drug interactions. So the basis of this work was from one of those papers which reports there is a increasing prevalence of diabetes in India, irrespective of social circumstances of the population, and the most common ones being obesity and insulin resistance. And we had actually taken up two drugs. We had taken glibenclamide and metformin. Glibenclamide was presented in another conference, so I chose metformin for this presentation. The evidences of herb-drug interaction are cited: affecting absorption, cilium inhibits lithium absorption, then meadow street, black willow, they displace highly protein-bound drugs like warfarin, licorice decreases metabolism of corticosteroids, there could be an additive activity like valerian increasing hypnotic activity of benzodiazepines. So the aim of the study is to assess the pharmacokinetic and pharmacodynamic interactions of metformin with Sallaki, which is known as Boswellia serrata, and Ashwagandha, Withania somnifera. Now, why did I choose Sallaki and Ashwagandha? Ashwagandha is taken for overall well-being. It is an immunomodulator and it is for longevity. Sallaki is taken very popularly because it is used for chronic inflammatory conditions. So that was the basis that why we had selected these two drugs. The objectives, we had to devise a bioanalytical method development and validation for metformin. Then To mimic that impaired glucose tolerance, we had to develop and optimize an animal model for insulin resistance, where we had to give different diets like high fat diet, high sucrose diet, and then we had to see which one of the best suits the criteria for the impaired glucose tolerance and hyperinsulinemia. Then, after which assessment of influence of Sallaki and Ashwagandha on the PK and PD of Metformin was assessed. This is the chromatographic method where the bioanalytical method for Metformin estimation was carried out. I'll not go into the details. This is the representative chromatogram for the Metformin estimation, signifying the lower limit of quantification and the upper limit of quantification. This again represents the calibration curve of Metformin. The slope you see here, the slope is close to 1. Then these are the important parameters, the percentage coefficient variation and mean percent nominal concentration, which is 14.76 and 104.21. And this is the internal standard that we used, atenolol. Then these are the stability parameters of Metformin. Now coming to the optimization of insulin resistance model. The background is type 2 diabetes mellitus is a condition which is characterized by insulin resistance. There's impaired glucose tolerance. There's hyperlipidemia. Then there's a fatty liver. So we need to simulate these conditions in the rats or the rodents. The epidemiological evidence says that most type 2 diabetic individuals are generally obese and have abnormal lipid profile, and this could be because of sedentary lifestyle and changed food habits. So we had to develop a model which could similarly or mimic those conditions that we see in the clinical side. So representing the conditions prompting the use of metformin. Now why do I say this is because metformin is not a hypoglycemic, it is an anti-hyperglycemic. Whereas glibenclamide is a hypoglycemic. So that was the reason why we chose metformin, because we had to see that how does it bring down the raised glucose levels. So for this, we had referred these 4 papers, Srinivasan et al., Jang et al., who have used different models to induce IGT, impaired glucose tolerance, out of which, with suitable modifications referring to these papers, we had an in-house diet. The composition is represented here, the high fat, the high carbohydrate diet, the HCD. Then we had these groups, normal control, which received NPD. NPD stands for normal pellet diet. HCD, high carbohydrate diet, which received HCD and water. HCD sucrose 30%, HCD fructose 25%. Then we used STZ1 at a dose of 35 mg per kg and 20 mg per kg. Now this was done, these groups were done to find out which animal model suits the best. So accordingly you see the food, water and body weight, it significantly increased in the high carbohydrate diet model. So we were able to establish one parameter, that is the body weight increased. So it could be leading to obesity. Yeah, you can see here the body weight drastically increased in the HCD, the high carbohydrate diet model. Then as per the OGTT excursion, you see here the normal pellet diet over a 15, 30, 60 and at 120th minute. And then HCD plus sucrose, it raised after 30 minutes, 1 hour and close to 2 hours. So this, I'm sorry for this graph. So this is the OGTT for HCD and this is for the HTZ. So looking at all parameters, we went for the high carbohydrate diet model. The blood sample was collected at 0.51, 2.53 and 24 hours in microcentrifuge tubes containing K2 ADTH. Samples were centrifuged at 3000 rpm for 15 minutes. Plasma was separated and frozen at minus 20 degrees. Then the samples were used for metformin, not for glucose estimation, and analyzed by HPLC method. So here, now here we had 5 groups. The NM were normal rats receiving metformin, NMA, normal rats receiving metformin and ashwagandha. Here, normal rats which were pre-treated with ashwagandha and then received metformin. NMS, normal rats receiving Sallaki alone. And then we had the other one where we pre-treated the animals with Sallaki and then gave metformin. So there are 2 things here. Groups where the animals were pre-treated with Sallaki and ashwagandha and then given metformin. The other side of it is simultaneous administration of metformin and Sallaki and metformin and ashwagandha. So you see here, in this group where it was pre-treated, the AUC and Cmax have significantly increased. Similarly, in the pre-treated Sallaki groups too, the Cmax and AUC have significantly gone up. Take a look at this data. There is a profound increase in the bioavailability with the pre-treated ashwagandha and Sallaki groups. This curve shows for normal metformin, which means, don't you think that there is a need to understand why these drug interactions are occurring? So this clearly says that in presence of ashwagandha and Sallaki, something is happening. See, you see this? There's an exaggerated increase. Similarly, the slide which I showed, that was for the normal groups. Now here, these are groups where the D stands for diabetic. The diabetic mellitus group animal, the animal which received ashwagandha in diabetes mellitus, then pre-treated with ashwagandha, only Sallaki in the diabetic animals, and the last one, pre-treated with Sallaki and received metformin. Here again, we see that the IR stands for insulin resistant. Look at the profile of metformin in the insulin resistant animals, and here there is a significant decrease in bioavailability with Sallaki pre-treated groups, which means it can affect the bioavailability of metformin. And which herbal drug is doing that? That is Sallaki. So similarly, we see there is an exaggerated curve here with ashwagandha, here metformin, and here pre-treated Sallaki stands different. All others are above the normal AUC. These curves represent the same thing what I had shown in those pictures. And again here the 12-hour plasma glucose profile post-metformin treatment in the insulin-resistant rats. Here, metformin alone was comparatively linear when compared to the exaggerated ones with the animals that received ashwagandha alone, animals which were pre-treated with ashwagandha. This is the same thing which is in a bar diagram. So the key points of the discussion is drug exposure in insulin-resistant animals was much higher compared to normal animals. There was a significant threefold rise in Cmax and AUC values in insulin-resistant rats. The systemic clearance was significantly low. T-half was prolonged. Indication of increase in metformin absorption due to pathological status is evident, and that was when we went through these 2 papers, Jang et al. In fact, in this Jang et al., he represents that model which was used with suitable modification. And a clinical study also shows that there could be a organic cation transporter expression in adipose tissue which could affect the distribution of metformin. Prompts for consideration of extent of obesity to individualize the metformin dose in those people who receive or who take Sallaki along with metformin. Effects of single dose of metformin in insulin-resistant rats, generally the action is slow and it requires several days of treatment. So our observations in the study was the mean plasma glucose levels of diabetes mellitus group were either maintained at baseline or they were lower than the baselines. in spite of access to food. There was no hypo or hyperglycemia, and it was not observed, and this condition was not observed in any other group. Then I had shown you the picture where we had shown those chromatograms, the LLOQ and ULOQ, where there were double peaks. Now the presence of double peaks was observed in the metformin profile. Now that is because it belongs to the BCS class 3 group. The BCS class 3 where metformin is highly water soluble but poorly permeable. And this could be attributed to alteration in the intestinal transit time. And one interesting observation in our study was this double peak phenomenon was less evident in the insulin resistant tracts. So this supports the OCT1 expression of Zhang et al.'s paper. And possible obesity could make metformin kinetics more predictable. Okay. How many minutes do you have?
Already in the next.
Okay. I'll quickly. Okay. So it did not affect metformin kinetics. The PD data did not show any observable difference, and the pharmacodynamic data was unclear. The insulin-resistant animals showed abnormal rise in plasma glucose post-food intervention. So, to conclude, increased obesity may cause increased systemic exposure of metformin. Metformin demonstrated predictable plasma glucose profile in the absence of herb treatments in insulin-resistant animals. And Ashwagandha did not affect the kinetics, but it was Sallaki that significantly reduced the bioavailability of metformin administration. I acknowledge Manipal University, Manipal College of Pharmaceutical Sciences, and Akron Ekinova. And these are my references. Thank you.
Thank you for this presentation. Have you any questions?
Quick.
I have. Which part of the Sallaki and Ashwagandha you have used, and
It is not the part. We had procured the pure extract of Sallaki and Ashwagandha from Himalaya company.
it is prepared on?
It is the pure extract which comes with the authentified COA, the certificate of analysis. It is not the leaves or the roots. If you ask for the pure extract, you get it from as a gift
Solvent
from
system.
solvent. No, we haven't gone into the solvent system of this because we have not gone for the extraction of
Okay.
Sallaki. You get it as a pure dry extract. Thank you.
Have you done some kind of a survey in which you have found out whether actually patients are taking these two herbs?
Yeah, we have done that. We had gone through a paper. I didn't do a survey.
Okay.
There's a paper by Adrian Few Berman where he has reported that out of 1,000 patients who have been admitted in the hospital, they have taken drug interactions more close to 537 were reported, and that was more due to St. John's wort. St. John's wort,
Yeah.
but these two are also on the list.
Yeah. Means what I meant to say was that whether actual patients of type 2 diabetes are taking these two
Yes,
herbs.
they are.
And
Because the reason is, sorry to interrupt you.
No problem.
The reason being, Sallaki is popularly used because it is used for chronic inflammatory conditions. Sallaki is popularly used for osteoarthritis. So there are people who are suffering from diabetes.
So maybe the exhibition is different, but they are
But
using it.
it becomes a common option.
And do you want to follow this up clinically also?
Yes, we do.
As some kind of a prospective or a
That-
retrospective study?
That's what we actually wanted to go for that. In fact, I had a few slides, but just for want of time, I couldn't present it. So those are our future directions. Thank you.